Over the next 45 minutes or so, we will be sharing important updates on the early detection competition we ran in 2024, share a video highlighting one of the innovative projects that was awarded funding, and welcome lead researcher Dr. Christopher Mueller and his graduate student Sydney Shepard to answer questions about their funded project. Please enter your questions using the Q&A chat function as mentioned before, and we will do our best to get to all of them following the video. But first, I'd briefly like to set the stage about why early detection research is so important. So the rationale for early detection is quite clear, as 5-year overall survival rates for ovarian cancer vary by stage at diagnosis. It is important when looking at these statistics to remember a few things. First, they include all kinds of ovarian cancer. Second, survival estimates are based on the experience of groups of individuals and cannot accurately predict a particular person's chance of survival. That said, the chance of surviving for 5 years is around 90% for those diagnosed at stage 1 and around 70% for those in stage 2, as indicated by the darker teal symbols. Unfortunately, we all know that 75% or around 75% of patients are diagnosed at stage 3 or 4 when survival rates are not as high. The challenge, therefore, is to develop ways to shift diagnosis to an early stage when treatments are more likely to be effective. If this was possible in most patients, it would lead to substantially improved survival rates for ovarian cancer overall. The issue, of course, is that detecting ovarian cancer at an early stage is very difficult. So, why is this? First, although most people diagnosed with ovarian cancer experience one or more symptoms leading up to their diagnosis, these are typically common, vague, and nonspecific. Examples can include persistent bloating, feeling full quickly, changes to urinary and bowel habits, menstrual changes, abdominal discomfort, or fatigue. Unlike some other cancers, there are no specific red flags for the individual experiencing the symptoms or for their healthcare provider. Secondly, there are significant technical challenges to detecting ovarian cancer at an early stage. Current diagnostic tests like transvaginal ultrasound and the CA-125 blood test are not sensitive or specific enough to accurately detect precursor lesions or small cancers. Also, until the discovery of the role of the fallopian tube in high-grade serous ovarian cancer, imaging tests that were focused solely on the ovary were simply looking in the wrong place. Finally, the biology of ovarian cancer is complex. There are many different types with distinct tissues of origin and clinical behaviors. This makes a one-size-fits-all solution for early detection very difficult. As an example, high-grade serous cancer, which again accounts for 90% of ovarian cancers diagnosed in stage 3 or 4, predominantly originates in the fallopian tube and can spread when the original tumor is still very small before symptoms develop. This is in stark contrast to other types of ovarian cancer, which can develop right from the ovary and enlarge the ovary as it's growing. So, to sum up, detecting ovarian cancer early is a complex challenge and also a top priority for our community. The good news is that science and technology have advanced so much over the past 20 to 30 years that it may finally be within reach. It was this optimism that led us to launch a national competition in September 2024 with the goal of funding 2 research projects with the greatest potential to drive advances in the early detection of ovarian cancer. Because of the failure of large screening trials to date, we challenge researchers to come forward with innovative ideas that leverage the crucial advances in both scientific understanding and technology over the past few decades. Application from researchers from across Canada were reviewed by an independent international committee of expert scientists, clinicians, and patient advocates. 2 exciting projects were awarded funding. The first, fallopian imaging and lavage for early stage tubo-ovarian cancer detection, is being led by the early career clinician scientist Dr. Andrea Nielsen from the BC Cancer Center. We will share a link in the chat if you want to learn more about this project. The second, and the highlight for tonight's session, is the earlier detection of ovarian cancer using an mDETECT liquid biopsy. This is being led by Dr. Chris Mueller, PhD from Queen's University. While funding these projects is only the first step, we are very excited to watch them progress and see the impact that they may ultimately have. With that, I'm thrilled to share with you a video featuring Dr. Mueller and Sydney about their mDETECT project. They will then join us live to answer questions.
When I was first diagnosed, I was in shock, devastated, and I think the strongest feeling that I had was feeling a loss of control over my own body and my own health. And later on, feeling like a whirlwind of how did this not get detected sooner?
Typically, it takes about 6 months to a year for a woman to be diagnosed with ovarian cancer from the first time she shows symptoms or the first time she has contact with her doctor.
Right now, ovarian cancer is diagnosed in those later stages, and that's one of the big reasons why ovarian cancer is such a deadly disease.
It's important to invest in early detection so that if you have early detection, then there's better chance to help people's health.
Ovarian Cancer Canada is a great partner for us because they're providing us with not only funding, but also patient perspective, that larger sort of global perspective on how our research fits in, really context for what we're doing. Now, ovarian cancer is really a problem, and it's different than other cancers. So often, if you develop cancer, you have a cough, you have a pain somewhere. We can, use imaging to actually identify where it is. The problem with ovarian cancer is that its symptoms are very diffuse.
One of the things I've heard from a lot of the patient partners that I've worked with is that they often had symptoms. They had bloating or some pain, and it was often misattributed to either menopause or other abdominal conditions.
One of the things that surprised me is how few women actually knew how prevalent ovarian cancer is and how there is no detection test.
So it can be very hard for them to come forward and basically have their physicians recognize that as sort of the pre-symptoms of cancer. And so it's really a situation where it's difficult then to enter into the process of actually being diagnosed. Worse, though, is that the process that we use for diagnosing ovarian cancer is actually not very efficient. So it's unfortunately the perfect storm of non-specific symptoms and difficult to diagnose. So that's why ovarian cancer is almost always diagnosed late. And that's really the problem with ovarian cancer, is that late diagnosis. Because of course, the women who are diagnosed late, they have a much poorer outcome. They have more symptoms, the disease is more difficult to treat, and more of them die. This project really comes out of research that we've been doing on blood tests for cancer. And it's a very specific test that we've developed for other cancers, but now applied to ovarian cancer specifically. And what happens is that when a cancer grows, a lot of the cells are actually dying. So even though it's growing rapidly, many of those cells are dying because the blood supply is very limited, and so the cells can't get oxygen or nutrition. So they die, and when they die, the DNA from those tumor cells actually gets released into the bloodstream. And what we've done is we've developed a test that actually detects those little bits of DNA from the tumor that are actually in the circulation. So when we take a blood sample out of a woman, we can look at that sample and say, okay, this particular DNA molecule actually came from a tumor, and say, okay, that woman has ovarian cancer based on this signature that we can see in the blood. One of the advantages of our test is that it actually is quantitative. So we can say, how much tumor does that woman have? Is it early on? Is it later? And particularly if it applies to their treatments. But it's really the early diagnosis that's the most important aspect here, because the disease is so much easier to treat when it's caught early.
Having the patient perspective could change something in how an early detection solution could be approved and adopted. Most screening programs aren't perfect, but having an early detection screening solution for ovarian cancer can save lives.
We know we can detect ovarian cancer in women who are diagnosed. What we don't know is whether we can actually detect it in women who haven't been diagnosed yet, but are undergoing that process. Funding from Ovarian Cancer Canada has really allowed us to take that next step, which is actually to look at patients, to allow us to go into those hospital settings and actually approach those women and get samples from them so we can actually test it in real life. So it's really about taking it from what's a laboratory-based experiment out into the real world where we can actually find women who are being diagnosed and actually validate the test there.
The reasons why I wanted to participate in this particular project is really so that other women don't have to go through what I went through and what a lot of other women go through. And then I have a 23-year-old daughter, so I obviously think of her.
In order for our test to be acceptable, we really have to look at how it works. Does it perform the way we expect it to? Does it have the characteristics that we expect? We've done the validation in women who have cancer. So we know that, we take blood from women who have ovarian cancer, we can tell they have cancer, and the signal we get is very high. But the women that we're actually dealing with in this study have all sorts of other issues, benign growths, endometriosis, they could have a whole lot of other things. And this is why they're presenting with these non-specific symptoms, because they're overlapping with the symptoms for ovarian cancer. So the question really is, can we not just detect ovarian cancer, but can we tell it apart from the things that these women are coming in for otherwise, that are basically hiding ovarian cancer? And so validating this is really very important for it to be something that can then be used in the public.
OCC has been an instrumental part to the work that we've been doing. The first thing that we did with some of the funding from OCC and their support is some final validation and some patient samples that were banked. And one of the big things we've been working on is the ethics approval to get our clinical study started. And we are going to be doing it over 2 years. So what's going to happen is women are going to come into the hospital with a suspicious ovarian finding, and they're going to be worked up throughout their standard diagnosis process. But when they first come in, we're going to be taking a blood sample from them, and we're going to see, can our test detect the disease, and we're going to compare it to what the current standard is. One of the other things that we're hoping to work on is look at treatment monitoring for these women. So once these women are diagnosed, are we able to monitor their treatment to make sure that it's effective? And if it's not, we can start making clinical changes. So overall, we're hoping that this test will be able to help women throughout the entire ovarian cancer patient journey.
Our early results really have shown that we can detect ovarian cancer extremely well. So in these women who have a diagnosis of cancer, we really can determine their levels are very high, which is unfortunate because it really indicates this is part of the problem, is that their levels in our test are almost the top of our range. So it indicates really that we have enough sensitivity that we can really detect women with much smaller tumor burdens, so much less tumor that's present in their body.
This really big difference between the two groups is letting us have a very accurate test. The really exciting thing is that we can go even earlier when the outcomes could be a lot better for these women.
A study like this, the focus is to find an acceptable screening process and get that approved by Health Canada and elsewhere in the world, and that really is the hope for the future young women out there.
I'm really proud to be part of the Dawn Ride, raising funds for this specific research. We get to learn about the exact project and progress of the project, and hopefully we'll be part of the project till the end, and we'll be able to see this early detection research come to fruition so that children don't have to grow up without mothers, and mothers don't have to lose their daughters.
The goal here is really to make a difference in women's lives. Ideally, we would use this as a screening tool, like women go for mammography, so we could say, okay, women over 50 or who have symptoms, a simple blood test every year or every 2 years could help them basically determine whether they have ovarian cancer, just like we do for mammograms. This research really matters because it can change the trajectory of a woman's disease with ovarian cancer. And it's really how we turn something around, how we go from a disease that is almost always fatal to a disease that is actually easier to manage and treatable and curable.
It's my pleasure now to introduce our guests. Dr. Christopher Mueller is a senior scientist and professor at the Sinclair Cancer Research Institute at Queen's University in Kingston, Ontario. His research has involved studying epigenetics in different cancers, the relationship between stress and BRCA, and developing methylation-based liquid biopsies. He earned his PhD in microbiology and immunology from McGill University and has received a variety of grants for advancing mDETECT assays to clinical validation in lung, breast, prostate, and ovarian cancers. He co-founded mDETECT in 2022 and is involved in all aspects of company growth. Our other guest, Sydney Shepherd, is a PhD student in the Department of Pathology and Molecular Medicine at Queen's University. She completed her undergraduate degree in life sciences at Queen's, where she specialized in cancer research. Her current research in the Mueller lab focuses on optimizing and validating a blood test for the improved detection and monitoring of ovarian cancer. So thank you both for joining us. And also would like to shout out that Chris is, I think he's joining us at midnight his time. So thank you as well for that. So as you know, early detection is a very high priority for the ovarian cancer community. So we're very appreciative that you have agreed to join us today. So there are a lot of questions, but I think I'm just going to start with one simple one. And it will kind of allow you to elaborate on a few key points about your study. What defines success for your study? What are you hoping to see tangibly through the lab work and through the clinical trial?
Absolutely. And thank you so much for the invitation. I really appreciate it. And of course, the support from Ovarian Cancer Canada for this project. Success for this project in particular really means validating our test in a real world population. You know, as basic researchers for many years, we've really, you know, we approach problems that are very well defined. We research them in the lab, we do very controlled experiments, but then we have to face that sort of aspect of going out into the real world and seeing, does our expectations actually meet reality? And so in this particular case, it's all about identifying whether the patients that we're looking at really do have ovarian cancer or not, and can we differentiate them from patients who have other symptoms. And so that is really the goal of this overarching problem, but really sets the stage for the next level. So how do we actually get this out into the public? How do we translate this into something from the laboratory, into something that has a real world aspect to it?
Thank you so much. Sydney, jump in if you want to add, or I'll go to the next question. So we have a question from Sarah, who's very enthusiastic. I've looked into this mDETECT study before, and it's so exciting. Question, can it be used to detect early recurrence if we finish treatment and told we were NED? Can we ask our oncologist for the testing if in Ontario? So maybe that's a little premature, but
Right.
just if it was successful, is this something you see as use of the mDETECT test?
Absolutely. So this study particularly is about early detection, but we have a lot of experience with our other tests in both lung and breast cancer, where we really then use the test to follow patients through their ongoing treatments, or if they're finished treatments, to monitor them for relapse and progression. And that's been very successful. And typically, you know, I can say from our lung cancer tests, we can see things 6 months or more in advance of CT scans. And that's really, you know, the same test. It's the same technology that we apply to early detection can apply to, as Sydney described in the video, to the entire journey. So through their actual treatments, so determining whether the treatments are working, and then, you know, off treatment, have they progressed at some point? And we really feel that it offers a lot of advantages in terms of monitoring, but also being very specific for that use case. So yes, absolutely. You know, with the caveat that we're still, you know, probably a couple of years away from actually offering it to the public. So really, this is something that, you know, is occurring in context of trials. And we're trying to get it going as fast as possible, but it takes time to get these things actually out to the public.
Thank you. So there is a technical question, but I think it actually leads into a nice opportunity to differentiate from what is currently available clinically. So the technical question was, what type of DNA modifications are you detecting in the blood of ovarian cancer patients? So maybe perhaps you can compare and contrast to the CA-125 blood test.
Yeah, I'll turn that over to Sydney.
Yeah, thank you so much for having us. Um, yeah, so similar to CA-125 in that it is a simple blood test. What we've seen is that our test is a lot more accurate than CA-125. So CA-125 is often elevated due to other conditions. So it could be menstruating or inflammation, causing it not to be as accurate as some other tests. So in that sense, our test has shown to be more accurate. Our test specifically is looking at DNA methylation. So these are those epigenetic changes in the DNA. So our test is looking at a very specific regions of your DNA and looking for almost these like little flags on the DNA that are telling us that there's a cancer signal or not.
Perfect. So someone just put a question that is actually quite similar to a question I was going to ask as well. Um, could so always the caveat of if successful and your vision, could mDETECT be used as a screening tool in people without symptoms? Can you explain the difference between using it as a diagnostic tool as you're testing it right now in people with symptoms versus screening asymptomatic individuals? And someone specifically wanted to know if this would be relevant for the BRCA1 or 2 positive population, but you can also extrapolate your answer to the general population as well.
Yeah, absolutely. And so, one of the reasons we actually developed these tests is because I have a long history of studying BRCA1 and 2 genes itself in terms of, particularly breast cancer, but obviously its impact on ovarian cancer as well. And so when we designed it, we really had that patient population in mind. So, as Sydney can attest, she did a lot of work to ensure that BRCA1 patients were sensitively detected by this test and really were able to then benefit from it in this case. And when it comes down to that sort of diagnostic versus screening tools, of course we're operating in an area where in terms of diagnostics it's actually easier to do. Like the study that we're going to be carrying out at Sunnybrook and Kingston General Hospital, we expect 20 or 30% of the patients to be positive for that because they've presented with symptoms. And so it's still complex to determine whether they're actually diagnosed with ovarian cancer or not, but it's a population that's definable and has a higher frequency. It becomes more difficult when you go into a screening context. Then, we're used to thinking about screening in terms of breast cancer. Mammography is very useful tool. It's very effective, but it's partly been a success because breast cancer has a higher frequency. It's significantly higher than ovarian cancer. So it is in a way more worthwhile for the healthcare system to employ that in detecting breast cancer because of that frequency. But I think we really foresee the ability to use our test for women who are not necessarily going through the same diagnostic processes we're talking about for our current study, but who are coming in for investigations of some sort. So I think it's going to be a long time before we do this as a general screening tool. But if we can get the cost down low enough, I think it would be something that could be done in that context. But I think the next level is to really see women who are earlier or much earlier in that process. So, first contact with their doctor, you go for a test and say, okay, let's eliminate ovarian cancer first, and then start the workup from that point.
Perfect. So you did mention cost, and someone did ask, what is the cost? I guess maybe what is the current cost, and what cost are you trying to work down to make it more accessible?
Yeah, so this isn't being offered as a commercial test yet. I mean, that's, our company mDETECT, that's the reason we formed a company, is to really sort of bring this to the market. We expect it to be in, several $100s for a test like this, being run on a large number of samples. So something that, would not be a burden to the healthcare system. A lot of tests that are being offered in the United States, they're several $1,000s for tests, and there's many of them available not specifically for ovarian cancer, but for other types of cancer. And they're, you know, in the $1,000 to $3,000 range. We expect to be in the several $100 range. So quite accessible and hopefully paid for through the healthcare system.
That's great. So I have two questions that are to give a break from the hard science, perhaps. Um, can you elaborate? We've been talking a lot tonight about the importance of meaningful collaboration with patient partners, and throughout the research process. I know that you have done this. You've been collaborating with patient partners. So perhaps you could just elaborate a bit on how you are doing these collaborations throughout your study.
Yeah, Sydney?
Yeah, sure. I'll take that one. So we've been fortunate enough to work with several patient partners, specifically a support group in Kingston, Ontario called the Ovation Circle. So I've had the chance to speak to a lot of the women from this group to hear about their lived experience with ovarian cancer. And it's really been instrumental to what we've been doing because they've given us their lived experiences and their priorities and helped us sort of shape the research that we're doing. And one of the big things that we heard from a lot of these women, which again, I think I mentioned in the video, was that they had symptoms that like a year before their actual diagnosis that ended up being dismissed and not diagnosed till much later, which has really helped us shape this study and our future studies to hopefully use this blood test at the onset of those first symptoms.
Thank you, Sydney. That was
And
great.
I just want to mention, you know, I really appreciated Shannon and Karen's comments about, you know, the whole issue, you know, the issue of being an ovarian cancer patient and the fear that that engenders. And I think, you know, these conversations we've been having with people with ovarian cancer, women with ovarian cancer, is that, you know, we're acutely aware of how sensitive a period this is, especially the patients, you know, we're bringing into our study, where, you know, they're at fear of having cancer. And so that can often lead to, you know, people even not wanting to undergo testing. So we're very sensitive to, you know, how do we build our studies so that women are willing to do this, and, you know, eventually when we get it out and hopefully, you know, more generally available, how do they have that confidence in accessing that process? And so we're really very cognizant of those issues and you know, appreciate those perspectives.
Thank you so much. I have another question for Sydney, not to put you on the spot, but as a trainee, what drew you to this field? What drew you to ovarian cancer research and inspires you to continue this
Yeah,
work?
I've always been interested in working in the cancer research fields since a young age. And specifically when I got the chance to work on ovarian cancer, I feel very fortunate to be working on this project because it is, you know, a woman's disease and it has been underfunded and under-researched. And, you know, ovarian cancer is a silent killer. And we haven't seen much improvement in survival in the past like 60 years. So to be able to even contribute in a small way to this is something that I feel very fortunate to do. Especially this project going from the lab to the clinic and getting this test out to real women is something that I am very excited to be working on.
Thank you, Sydney. We're happy to have you in the field. So back to the nerdy science question. Someone asked, and maybe it's just a general question, how is the mDETECT liquid biopsy different from other circulating tumor DNA blood tests that are being tested as well? This person specifically called out Signatera, but you can make that a more general answer.
Sure, absolutely. And, you know, there are a wide variety of tests available as liquid biopsies for detection of various cancers. And some of them work well. A lot of them don't work that well. Most of them are based on mutations, identifying mutations. So when a cancer forms, there are specific changes to the DNA that occur. And those mutations, in many cases, are what drive that cancer. So they change a gene, it becomes more active, and the cancer develops. There are also other changes, the sort of epigenetic changes that we talk about and that Sydney mentioned, where it doesn't actually change the sequence of the DNA, it just basically puts on little modifications. And the real advantage of what we're doing with epigenetics and DNA methylation is that it's much more frequent. And so we can build a test that is actually probably 10 times more sensitive mutation-based assays. Mutation-based assays as well, in some cases, they actually need a sample of a tumor before they make a sort of specific test for that cancer. And that's a very long process and obviously requires access to material, tumor material, which for ovarian cancer screening is impossible. So the way our test works is really looking at a large number of these changes across a cell's DNA. And so it really results in something that's very sensitive and specific. And one of the things about our test particularly is that it's very specific. So Sydney was alluding to the fact that we have very little background. And so that's one of the advantages of a diagnostic test like ours and even a screening tool is that people who don't have cancer have very little signal from our test. And so it really places us in a position of being able to do the test with confidence and detect it much earlier at the same time. And so one of the other things is, obviously cost is a driver. So something like Signatera assay, which requires typically patient samples, again is $3,000 to $4,000, where ours is much simpler. It's concentrated on ovarian cancer specifically, and so can be offered for a much lower price.
Thank you. That was really informative. There's another question that just came in that I was hoping to ask as well. So obviously there's lots of different types of ovarian cancer. Is this a test that, again, if successful, is it designed to detect all types of ovarian cancer, or is it specific to certain types, most likely high-grade serous, or could it be used for some of the more rare types of ovarian cancer?
And we'll get Sydney to answer that. It's her specialty.
Yeah. Yeah, so originally when we designed this test, we did focus on high-grade serous ovarian cancer, as it was the most common and the deadliest form of the disease. So the original design was for high-grade serous, and a lot of the validation that we've done is for that subtype. But we have looked bioinformatically and seen, does our test work for some of those rarer subtypes? We've seen very, very promising results. Showing that not only could our test detect high-grade serous, but it could also detect those rarer subtypes. And throughout this study that we're doing, we're going to be taking blood samples from all women coming in with these suspicious ovarian findings. So there is a chance that we'll be getting samples from some of those rarer subtypes. If not, in the next little bit, we'll be getting samples from some of those rarer subtypes to validate the signal that we've seen, to ensure that it would work across a variety of subtypes.
That's great. I also want to talk about stage. Obviously, early detection is really about getting that stage 1, sometimes stage 2, but really the holy grail is stage 1 or stage 0, even with stick lesions. Have you tested the ability of mDETECT to detect ovarian cancers at all stages?
Yeah, so the test so far, we've been validating it on samples from biobanks. So these are samples that have been banked, and we're able to grab those and use those for our test. And since ovarian cancer is often diagnosed in those later stages, stages 3 and 4, that's sort of the majority of the samples that are available. So that's what we've mostly tested on. We have been able to test on some samples from earlier stages, and have shown good results in those earlier stages as well. But we're hoping with the study to be able to get some of those earlier stages and to continue to validate that signal.
That's
Yeah,
great.
the irony here is that we haven't been able to get samples from stage 1 and 2, unfortunately.
Yeah, and I wonder, I guess this is going in the weeds, but in terms of being able to access banks that exist that collected samples in hopes of doing things like circulating tumor DNA studies, if you could lean on those national resources. So perhaps just email me and I'll see if I can link you to some earlier stage cancers.
Absolutely.
So I think, okay, so this is a general question. How does someone with symptoms get flagged for the study at a hospital level? Of course, this is only happening at 2 hospitals. What guarantees that is there that you are getting people early?
So the idea is not to get them early in this stage. It's really to identify them as they're coming in for a workup, basically for their symptoms. So these are, people typically seeing a gynecologic oncologist because it's obviously suspicious for ovarian cancer. So that's the stage where we have to go through that first before we can work back to earlier and earlier stages. And at that point, it's a different kind of study because this particular study, we're looking for 100 patients. As I mentioned, we expected maybe 30 of them will actually have ovarian cancer. When we move to the next level, when it's earlier in that process, so maybe women coming to a gynecologist for a complaint, then the incidences actually fall quite dramatically. So rather than doing 100 patients, we'd have to do probably 1,000 patients in order to have enough actual patients with cancer. So that's hopefully the next step and takes us that step earlier in terms of diagnosis, but something that's going to have to be a sort of different design in terms of the study itself.
Perfect. And I think our final question tonight, I've seen a lot of questions about when will this test be available and just how many years it takes to get to that step. Perhaps you can just generally explain if this specific part of the project is successful, what would be the next steps that you would need to take to make it more widely available clinically?
Yeah, so
Even in that more narrow context of diagnosis.
right. So really the next step is more patients. And that's where, we're trying to basically expand to multiple centers. Because there's not a threshold, but the more patients we have, the more experience we have with, different diseases, different underlying diseases that are not ovarian cancer. And so that's really how these tests get validated, because we have to demonstrate to, Health Canada, this is a test that is actually specific, because they're not going to approve it if it's not actually doing what we say it is. So 100 patients, it's a good start. Ideally, we'd like, several hundred more as the next step to actually be able to go to something like Health Canada to at least start the approval process. And there are processes for some, sort of urgent cases which you can build, which allow for more rapid approval as well.
Perfect. So I think, unless you have some final words that you'd like to share, first I'll give you that opportunity. If there's anything that hasn't been asked that you would like to ensure gets told to everyone first.
Yeah, I think it can be frustrating when research doesn't go fast. And, having been a researcher for 35 years, it does seem slow. But now that we've gotten to this stage, I think, there's hope. And this is where doing translational research is very exciting for researchers as well as for patients, because we are making that transition to something that is actually coming out to the real world. So I think, there are ways for us to accelerate that. And I think we're really committed to this and really seeing it come to market and be accessible as soon as possible. Sydney, do you want to add anything?
Yeah, I just want to thank everyone again for giving us the opportunity to speak about our research. And yeah, just want to highlight that what we've seen so far is very promising. And we're very excited to see the results of this clinical study to take that next step towards getting this available for everybody.
Thank you so much to both of you for joining us. The chat has been on fire, so clearly people are excited to hear about this. And again, we look forward to watching this progress and wish you all the luck with this exciting project.